This is Bernard Paulson, and this is the second lecture from the book indicated on this slide. What we will talk about are the basics of chemical kinetics. It is a topic that may be already familiar to many of you, at least some aspects of it. So the outline covers a few topics. First, I want to go over the fundamental concepts associated with dynamical analysis. I'd like to introduce the governing equations we use to simulate dynamic states of networks. These are the dynamic mass balances. In these equations, we have kinetic features showing up in the form of reaction rate laws. Since we are dealing with networks, we typically have many different timescales, and the models represented by a dynamic mass balance are multi-scale. Finally, I want to talk a little bit about the underlying assumptions in formulating the dynamic mass balances, and focused in particular on the constant volume assumption. So first up, the fundamental concepts. So there are four fundamental concepts that we need to be cognizant of in this field. First is the time constant. So the time constant is a measure of time, and we will see many different time constants in this class. The second concept is the notion of aggregate variables. When a multi-scale model unfolds in time, fast timescales relax, which means that the dynamic dimensionality of the description shrinks. What that means in turn is the fact that the independent variables you started out with in the beginning develop dependencies. So this means that some of the original concentration variables will now move in fixed relationship to one another, and this leads to aggregate variables or pooling them together. The third concept is that of a transition, and that is a question of how a system moves from one steady state to the next, and this can happen in different ways. Finally, the data that we get, the numerical data we get, is very voluminous and complicated, and we need to be able to graphically represent it so we can visualize and conceptualize the dynamic response of the system that we are studying. First up are time constants. So a loose and general definition of a time constant is as follows. A time constant is a measure of the time it takes to observe a significant change in a variable or a process of interest. The graph or the sketch at the lower half of this slide illustrates this. If we are tracking a variable x that changes by some magnitude indicated by delta x, and if it is changing at some characteristic rate as indicated by the average time derivative, then the ratio of these two quantities will be the characteristic time, or the length on the x-axis that is required to measure the duration of this change. This is the notion of a time constant. There are many, many ways in estimating a time constant or defining a time constant. Let me give you one example of a time constant that is important to all of us in our lives. And that relates to balancing a checkbook. In this case, the time constant is 1 month, and we may get our paycheck into our checking account every month. Then we have rate of use of this cash. If we use the money in exact balance with the rate, with the deposits that go in, we will have used up all the money that was deposited at the beginning of the month when the end of the month comes around. If we use less, we will have the green curve that's in here that will lead to savings or accumulation of balance in the account over time. And conversely, if our rate of use is too fast, then we have the red curve, which leads to depletion of the money before the end of the month, which leads us to borrow. Of course, we all want to have the green curve. Aggregate variables. So here's a little primer on pooling variables. What this slide shows are the first 3 steps in glycolysis. First, glucose is phosphorylated by hexokinase to form glucose 6-phosphate. This is a relatively slow reaction. It's followed by the isomerization of glucose 6-phosphate to fructose 6-phosphate. This is typically a very rapid reaction. The third step is then a second phosphorylation step by phosphofructokinase. That's also a slow step. So here we have 4 compounds, 3 reactions, and we've defined 2 different timescales. Timescale separation like this can lead to temporal decomposition. What that means is that the events in the red box are fast compared to the events that fill and empty that box, if you will. As soon as glucose 6-phosphate is formed by hexokinase, it's immediately isomerized and an effective quasi-equilibrium forms between the 2 hexose phosphates. This means that on a slow time scale, their movement will be indistinguishable, and they can be grouped together, pooled together to form an aggregate variable that is called hexose phosphates. And this is what's indicated on the lower half of the slide, where you have a slow input and a slow output from this red dashed box. There are events within that box, but they are too rapid to be of interest, and for all practical purposes, there is only one variable, one dynamic variable within the confines of that box. So that's the idea of pooling, and as a consequence of relaxation of fast dynamics and the development of dependencies amongst your state variables that you started out with. Transitions. This slide shows the transition of a system from state number 1 to state number 2. Something induces a change, and the system moves from one state to the next. How this transition takes place may vary. And there are 5 categories of characteristic transitions given at the bottom of this slide. The first one we call a smooth landing, which is the yellow curve, which may be similar to an exponential decay. Curve number 2, the green one, is an overshoot. In this case, the system starts moving from state 1 towards state 2, but it overshoots it, then turns around and settles down in state 2. The third curve is a damped oscillation, that is the red curve, and that's basically characterized by a series of overshoots that get smaller and smaller with each overshoot, and eventually the red curve dampens down in that steady state number 2. The fourth one is an example of a dynamic instability. State number 2 is dynamically unstable. So as state 1 is left, as the system leaves state 1 and heads towards state 2, it overshoots it, returns, overshoots it again by the same magnitude, and oscillates continuously around that steady state. That steady state is known as, is called to be dynamically unstable because the system will never settle down there. There is a fifth type of transition that's even more complicated than the sustained oscillation, which is a chaotic motion. The last 2 or even 3 of these responses transient responses are the subject of nonlinear dynamics, and they are studied by looking at the Jacobian matrix mentioned in the last lecture. We will not deal with them in this class. Most of the responses we will see represent type 1 or 2 transition as illustrated in this slide. Finally, graphical representation of the solution. We can now graphically represent a solution in many different ways. There is no single best way of doing it. There will be a suitable way for you to conceptualize or develop a particular system. So often you have to try a few of these candidate representations until you come up with the one that suits you in terms of understanding the response of your system in a given environment. This slide shows 3 types of graphical representations. The first one, A, would be putting little graphs as inserts into a network map. That is a complicated representation and hard to program. Most of the time you will be doing something that is shown in panels B or C. In panel B, we show the very classical way of showing a response, which is simply plotting a variable as a function of time. Sometimes we do variations on this representation, as you shall see later in the class, and one such representation is to put the variable x onto a logarithmic scale. And to the right, excuse me, we show a two-phase response on a log linear scale. Well, first there is a fast response followed by a slow response. And this is one reason for doing a log linear graph. And this, for instance, may represent the example we had earlier, where there is initially a rapid equilibration of the hexose phosphates into a pool, and then a slower dynamic that fills and drains that pool. The last panel on this slide is important. This is called a dynamic phase portrait. In this case, we plot 2 concentrations against one another and form a trajectory in this plane using time as a parameter. So the initial state will be a point in this plane as indicated by the point time equals 0. Then you will see a dynamic response unfold, and the point will move around this phase portrait and eventually settle down at another point where time goes to infinity. This turns out to be a very useful graphical representation. Our next topic is a discussion of the governing equations that describe the dynamic states of a network under the assumptions outlined in the last lecture. We start out by looking at the mass balance equations. This little schematic shows a concentration x sub I that is formed and degraded. It's formed by 2 different reactions and degraded by 3 different reactions. The symbols in here indicate flux. V sub a number like V sub 3 represents the flux through that reaction. X itself is in units of concentration. Discuss these a little bit more later on. The dynamic mass balance for x sub I is simply a summation of the fluxes going in to this node that x sub I represents minus the summation of the fluxes that degrade x. So in this case, we have 2 positive terms leading to the formation of x sub I and 3 negative terms that lead to its degradation. If the flows in and flows out, or the flux is in and the flux is out, they're perfectly balanced. We have a steady state, and the time derivative dxi dt is equal to 0. As I mentioned in the last lecture, this summation can be represented as an inner product. We have in this inner product a row vector, which will turn out to be a row vector in a stoichiometric matrix, and a flux vector, which is normally a column vector, but is transposed here to form a row vector. So you can readily see, if you know the laws of the inner product, that if you multiply these two vectors together, you get that summation shown right above it. So this is the dynamic mass balance on a single compound. We can write out a mass balance like this on every compound in the network that we have defined. When we do that, we get a matrix equation as shown over here. Now we have the time derivative of a vector x that has all these x sub i's as entries in it. On the right-hand side of the equation, we now have a matrix S, and every row of that matrix is the same sort of a vector as we saw in the previous slide. And then this matrix S, which is the stoichiometric matrix, is multiplied by a column vector of the reaction rates. The reaction rates that we will discuss in more detail later on are functions of the concentrations, x, and have a number of rate constants in them designated by k in this equation. Let's briefly review the dimensions of these quantities. First, the stoichiometric matrix is dimensionless. All the stoichiometric coefficients have no dimensions. The time derivative of the concentrations will have the concentration, which is normally moles per volume, in the numerator and time in the denominator. So this will be in terms of mass per volume per time or moles per volume per time, depending on the application. The fluxes, the V's, also are mass or moles per volume per time. The concentrations X are moles per volume or mass per volume. The rate constants that we will be looking at will fall primarily into two categories. There is a first order rate constant that has the units of reciprocal time, or a bilinear rate constant that has units of reciprocal time, reciprocal concentration. This is our master equation, and just by looking at it, you realize you need to know ordinary differential equations and linear algebra. You should have had both of these classes as prerequisite for this class, and we will be using material from those classes that you've had, and we will rely on your understanding of that material. And I will assume that you know it. If you don't, you should go back and review it early on in the term. Often, the difference between a reaction and a flux through a reaction is confusing. So let me try to avoid any such misunderstanding from developing in your mind. At the bottom of this slide, we show a matrix, stoichiometric matrix, times a flux vector. Let me make three comments on this equation. First, the columns of the stoichiometric matrix represent the reactions, and we use n, the index n, for them to designate the number of them in a given network that we're looking at. These are the stoichiometric coefficients. They tell you which compound is being converted into what compound. The rate at which that happens is the number contained in the column of reaction rates. So V is actually the rate at which that reaction happens. It doesn't change the reaction itself, what's being converted to what, but it tells you the the rate at which it happens. How many moles per volume per second are being processed through that reaction. And if you remember your matrix multiplication, you will see that this matrix representation here, when you multiply it out, will become the first column in that matrix times V1, the second column times V2, and so forth. So V1 will multiply all the elements of the first column the very same way. Now let's next talk a little bit about kinetics. This material should be familiar to most of you from a previous class. The master equation shown earlier has a series of reaction rates in them. The fundamental type of a reaction that takes place in a reaction network is either linear or bilinear. These are called the elementary reactions. The linear one takes a compound X and converts it to something else. A bilinear reaction results from the collision of X and Y. They react to form something. It is worth noting in these elementary reactions that the concentrations are non-negative quantities, and so are the fluxes. This means that the concentration or a flux through an elementary reaction is either zero or positive. They cannot be negative. Let me give you an example of a series of reactions. This is hemoglobin. Hemoglobin is a tetramer comprised of 2 alpha chains and 2 beta chains. The way it is assembled is shown with this reaction schema inside the red box. First, an alpha chain combines with a beta chain to form what's called a heterodimer of alpha and beta. Then two of these heterodimers react to give us the tetramer, the heterotetramer. So the assembly of this molecule is actually a series of 3 bilinear reactions. Sometimes they're all lumped together as shown at the bottom, where we say that 2 alpha chains plus 2 beta chains become that tetramer. We should all know that that is not an actual chemical reaction. There is no way that 4 chemical species, can meet at the exact point in space at the exact same time. Therefore, this is a lumped representation of the reaction. All the elementary reactions are either bilinear or linear as indicated above. We'll talk more about combining elementary reactions here in a moment. Before we move on, I want to talk about a special case. Sometimes there are reactions that lead to homodimer formation. So where 2 molecules react, but they are both the same. And they react with each other to form a homodimer. And that's indicated to the bottom right hand of the slide. I should say this reaction gives a rise to a stoichiometric coefficient of 2 in the stoichiometric matrix. The other reactions shown on here, the linear and the bilinear reaction, will lead to stoichiometric coefficients of unity. So let's talk next about the kinetics of how this happens. As you probably all recall from mass action kinetics, the rate of reaction, the rate of a chemical reaction, is proportional to the collision frequency of the molecules. Under the continuum assumption that we make, namely that we are not looking at individual molecules but at a continuum, the collision frequency of molecules will be proportional to their concentration, simply their count per volume, moles per volume. This means that the mass action kinetic description of a linear reaction is just a constant times the concentration of X, and for a bilinear reaction, it's a constant times the concentration of X times the concentration of Y. And if you think back a few slides, you will now see why the units on these two coefficients are different. As indicated in blue below, the assumptions shown on this slide refer to a homogeneous medium that is not restricted by its geometry. On occasion, the domain that we are looking at is geometrically restricted, and that may lead to power law kinetics as shown at the bottom of this slide. Namely, a first order reaction may not be k times x, but k times x to the a. And if collision frequency is restricted, that a will be less than unity. We are not interested in this detail of the kinetics, but it is important to be aware of the fact that we don't always have strictly mass action kinetics taking place. For all practical purposes, though, the mass action kinetics give us a workable description of the dynamic states of a network. Next, we will talk about the origin of the numerical value of the kinetic constant. Shown on this slide is the surface of a protein with binding sites for two molecules, A and B. After they are bound to the protein, they are found in a particular relative spatial orientation. And this spatial orientation will maximize the probability of a reaction taking place. If a reaction takes place in a solution, the angle of the collision between the two molecules determine whether you get a reaction or not. And this is illustrated at the inset to the left on this slide. A particular reaction, as shown on top, a particular type of collision, as shown on the top image in that insert, will lead to a reaction rate, whereas the type of collision shown at the bottom of the insert does not lead to a reaction rate. The rate constant is really a measure of the probability of the right collision taking place. So what determines the numerical value of a rate constant? The right collision does. The frequency of the right type of collision does. Therefore, K becomes a biologically determined variable because essentially all biological reactions take place on the surface of a protein. The surface of that protein is genetically determined and in an abstract way has information in it about the right angle in which these molecules should bounce off one another to have a reaction take place. The acceleration factor of an enzyme is a measure of the difference in the reaction rate of A and B reacting in a solution versus the reaction rate on the surface of a protein. Therefore, K is a biologically determined variable. It has a genetic basis. And in fact, we all have slightly different numerical values for our kinetic constants, since the sequence of our genes for the enzymes is all slightly different. This reinforces the comments I made about the contents of the gradient matrix in the last lecture. In blue here is a little sidebar for you to ponder. There are some interesting properties of protein that one comes across over time, and it's worth walking through them here perhaps. Most of the amino acids are chemically non-reactive in the sense of forming covalent and non-covalent bonds. Big exception from that is cysteine. There are, of course, some modifications on amino acid residues in protein that also happen, but typically the amino acid in a protein doesn't participate in the reactions shown above. So protein mostly work through hydrogen bonds attracting A and B to bind, and through the shape of their surface to make that proper angle form. Many of the aromatic amino acids and arginine have pi orbitals and therefore can shape the electron cloud on the reactants once they are bound to a surface to minimize activation energy and facilitate the reaction. Proteins are sticky. They seem to stick to everything except themselves. And then, as I mentioned earlier, phosphorylation and acetylation modification. Let me back up. The modification of amino acids on a protein by phosphorylation or acetylation change their properties and very often influence protein-protein binding that can activate or inactivate a protein. To get the right chemistry to take place on the surface of a protein, it very often needs a prosthetic group, a cofactor, a divalent ion, or something like that embedded in the protein itself to induce the chemical reactions. The next thing I want to talk about is combining elementary reactions. This is very frequently done in performing kinetic analysis. First, I'll go through this very simple example shown here. This is the reversible reaction of X1 being converted to X2 by an elementary reaction denoted by V superscript plus, plus meaning the forward direction of the reaction. And then there is the equal and opposite reaction when X2 is being converted to X1 through a reaction designated by V superscript minus. These are two elementary reactions that are working in exactly the opposite directions. Therefore, we often combine them into one net reaction, which would be the forward minus the reverse. Now, this net reaction rate, which is a combination of the two elementary reaction rates, can be positive or negative. If it is positive, the reaction is proceeding from left to right or in the forward reaction. If the net reaction rate is negative, then the reaction is proceeding from right to left or in the opposite or in the reverse direction. If the net reaction is 0, B plus and B minus are exactly equal in magnitude, and then we have equilibrium. So equilibrium are actually dynamic states with elementary reactions proceeding, but in exactly at the same rate. An equilibrium constant is used to describe the equilibrium. It is thermodynamically determined. It's a physical chemical quantity. Unlike the individual kinetic constants, this number is not subject to biological modification. It is determined by the thermodynamics. The combination of elementary reaction rates can get much more complicated than the simple example above shows. A classical reaction mechanism in biology is the Michaelis-Menten reaction mechanism. It's shown in the dashed blue box. Here you have a substrate binding to an enzyme reversibly to form an intermediate X, and that intermediate can then carry out the reaction, release the product, and regenerate the enzyme. So there are three elementary reactions in this mechanism. They can be combined under the quasi-steady state assumption or the quasi-equilibrium assumption to generate the well-known Michaelis-Menten equation, which is the rate law shown on the right. We will detail how this is done later, but I just wanted to show this to you now so that you know that the Michaelis-Menten rate law is actually a representation of many different elementary reactions under certain assumptions. The final quantity I want to introduce you to here is the mass action ratio, often designated by an uppercase gamma. Here I show the isomerase that we talked about earlier, where glucose 6-phosphate is converted to fructose 6-phosphate. If this reaction is carried out in a beaker, it will go to completion and go to equilibrium. If we can measure the concentration of the product and the reactant and take that ratio shown in the red box, you can experimentally determine the equilibrium constant. Now, if we have a closed open system, as I showed earlier, where there's actually a trickling in of glucose 6-phosphate and a slow removal of fructose 6-phosphate, you will have a system that will eventually reach a steady state. It is not equilibrium because we have inflow and outflow, and we will detail the difference between a steady state and an equilibrium state more thoroughly in a later lecture. We can measure the concentration of fructose 6-phosphate and glucose 6-phosphate in a steady state in an organism. If you ratio those two concentrations, we get this mass action ratio gamma. So gamma is an equivalent quantity to the equilibrium constant, but for an open system. And sometimes we take the ratio of gamma to K equilibrium and look at how close to unity it is. If it is very close to unity, it means that the reaction is close to equilibrium in the steady state. The next topic I wish to discuss is the multi-scale nature of dynamic models of biological networks. And to start this discussion out, I picked this example of the basic structure of high energy bond trafficking inside cells. We all know that ATP carries the high energy phosphate bonds. The molecule is shown schematically to the left of this slide, where we have 3 phosphate bonds bound to a pentose, and the pentose is also bound to a base, the adenine. ADP has 1 of these high energy bonds missing, and AMP has 2 of them missing, as designated by the empty open red circles. Now, if we look at this reaction schema, we see that ATP is used through a reaction V1 to generate ADP. V2 is the reverse process, so that's a process of making ATP. The reaction designated by V5 is a very rapid equilibrium reaction in which 2 ATP molecules react and exchange a phosphate bond to generate ATP and AMP. The carrier molecule, AMP, is synthesized by V3, which is a low flux reaction inside the cell, and then turned over by V4. It turns out that the concentration of AMP inside cells is very important because it's used as a sensor of the energy state of a cell. Associated with this system, there are a few important quantities. One is what we call the capacity to carry high energy phosphate bonds. And that will simply be the summation of these three forms of the adenosine phosphates. That's the capacity to carry high energy phosphate bond. So this total concentration will give you the volumetric density, the volumetric, the number of high energy bonds per unit volume that you can carry inside the cell. Now, how much of that capacity is actually used is defined by this occupancy parameter. There are two high energy bonds on ATP, one on ADP, and 0 on AMP. So I have now two quantities, capacity and occupancy. If I ratio those two, we get what is called the energy charge. This is literally like a battery. It's the capacity to carry charge and how much of that capacity is actually occupied. If we measure these concentrations, ATP, ADP, and AMP inside living cells, and carry out the calculation of the energy charge, we will find out that most of the time that it is between 85 and 90% as indicated. Thus, cells tend to be highly charged, if you will. This little insert here shows simply how this calculation is carried out. The capacity and occupancy are therefore combinations of concentrations, but represent combinations that are physiologically significant. Now let's look at the kinetic description of this system. What's shown here are the dynamic mass balances for the three concentrations involved in this little network: ATP, ADP, and AMP. The color coding, the dashed line under these reaction rates, designates the response times. So red designates something as very fast, blue something as very slow, and green something as intermediate. This represents a three-tiered timescale hierarchy. There will be slow, intermediate, and fast dynamics in this case. The structure of these equations are important because I can a priori do some pooling of variables here. Let's look first at the blue box. If I simply add these three differential equations together to get the dynamics of the capacity, ATP plus ADP plus AMP, you can see that the only fluxes that move the capacity are the slow fluxes, the fluxes that are the smallest in the system. So this capacity will, by default, have slow dynamics. If I look at the occupancy that I defined on the earlier slide, and I multiply the first differential equation by 2 and add it to the second one, I will cancel all the red terms on the right-hand side of these equations, and I get only the fluxes of intermediate response times to move the occupancy. So the capacity turns out to be a slow variable, and the occupancy an intermediate variable, and then there's a third fast variable indicated there that has limited physiological consequence. So the energy charge is a ratio of two variables, one that has intermediate dynamics, in fact represent the master timescale of energy metabolism, and a slow variable that is the inventory of, the capacity, the ability of the system to carry high energy phosphate bonds. So in general, we have a timescale hierarchy, and that's often indicated by drawing logarithmic scales, because timescales are often separated by many orders of magnitude. At the bottom of this slide, I'm showing this inherent system transients, and on the top of this diagram, we are showing the external timescales. We may be only interested in a part of this spectrum where we are observing a process or where the physiology takes place, and anything that's too fast for that, that is too fast relative to this timescale window, we can relax, and everything that is too slow, we can keep at a steady state. And some examples of that are shown here based on the example that I just gave. Finally, I'd like to talk about a couple of important assumptions. First is the constant volume assumption. When we draw a total mass balance on a compound inside a cell, we actually have to multiply the volume of the cell times the concentration. That will give us how many moles of a particular compound are inside the cell. I can do a total mass balance, dynamic mass balance, on a compound inside the cell, as shown in this equation, where the units of these fluxes are moles per cell per time, not moles per volume per time. Since the total mass is a product of volume and concentration, I can break that derivative up by a familiar formula shown below. And only if volume is a constant, I will have the time derivative of the volume being equal to 0, and I can convert the total mass balance into the volumetric mass balance that we showed earlier. This is the assumption of constant volume. There are other assumptions we make that are based on osmotic balance and electron neutrality. A cell, like the red blood cell, for instance, that is floating around in our bloodstream, experiences osmotic pressures. It has to balance osmotic pressure. It pumps ions across the membrane, like sodium and potassium, shown at the bottom of this slide, and therefore it has to balance electron neutrality because charges separate, elemental charges cannot be separated. So these are important constraints that are placed on dynamic mass balances that are actually often ignored for convenience sake. The volume assumption often leads to problems in model formulation, and here are a couple of examples, historical examples of bad assumptions. One is to build a cell cycle model on a constant volume, which many people do, whereas the volume is actually doubling. So a model like that cannot be realistic. Another frequent problem that occurs in multi-compartmental models is not to scale the membrane fluxes appropriately, as shown in this slide. If the surface to volume ratio for a shared membrane between two compartments is not the same, because the volume of the compartments can be different. In a few models in the literature, that surface to volume ratio is the same, and that leads to an error because the total fluxes in and out of these two compartments will then differ. And this leads to a famous statement that says hypotheses can be right or wrong, but models have a third possibility: they can be irrelevant, such as the two previous examples shown. Models are irrelevant if the assumptions made in the formulation are at variance with physical reality. This website here has some interesting information on it that you might wish to look at. So to summarize, the time constant is a key quantity. A spectrum of time constants leads to timescale separation and temporal decomposition. Multiple timescales lead to aggregate variables. Eliminating time constants lead to reduction in dynamic dimensionality and therefore to pooled variables. Elementary reactions are described by elementary rate laws that are comprised of non-negative variables. And finally, it's very important for us to understand fully the assumptions that lead to the dynamic mass balance equation that we use for all of our network models. I will not cover these extra topics here, but potentially will record them into a separate file for those that are interested in more advanced material. I thank you for your attention and hope that this voice over of the slides is useful.